So thank you very much, Francesca, and thanks to everybody. It's a great pleasure to be here, and thanks for this invitation. And I would like to tell you something about my activity and also to tell you something about my history as a researcher that started more or less around 2002 when I began my PhD. And I'm an associate professor in agricultural microbiology, so I am in that sector that is called in Italy AGR16, so I want to specify this. And all my activities are related to food, and also my department actually is working on food and everything related to food, from food production, food preservation, packaging, and everything that is connected to food and nutrition. And all my activity as microbiologist is connected to the relationship between diet and human health, as you can see here in this picture. And I'm anyhow studying microorganisms because what is actually very clear now is that if we do not consider microorganisms, it's not possible to understand properly how food and nutrition can have an impact on human health. And microorganisms have an effect in this kind of connection at different levels. And I'm speaking about microorganisms that are from the environment or from food, and also microorganisms which are constantly associated to the human body. And why they are both so important. Environmental and food microorganisms, it's very clear, we know they have an impact of food, and they can transform the food, they can change the availability of nutrients, they can increase, for example, the level of vitamins, and they can modify food. And for the main example, the most simple example, and well-known example, is represented by fermentation. Many food are transformed by microbes. And at the same time, it is also important the amount of microorganisms that we introduce with food, especially vegetables or other food that we used to consume as raw food. They are carrier of microorganisms and microbial cells. And especially this topic is quite probably unexplored. And it is the topic of my role as a microbiologist in a huge regional project that is called Mind Food Hub. I'm trying to understand how production procedures, so even farming procedures, can have an effect on the microorganisms vehiculated by, for example, vegetables, and also any kind of food actually, and how this can have an effect on the human-associated microbiota, mouth, gut, also vaginal mucosa. And then finally, probably the main part of my activity is related to this kind of role of microorganisms. That means how host-associated microbiomes can affect the effect of food on the human health. I will make a few examples on this. I will make a couple of examples of how it's so strictly important to have the right microbes in order to have the right consequence, the right effect once that we take a specific dietary pattern of specific food. More generally, so I'm working about microorganisms, all the possible microorganisms, even if one main experience on bacteria, related to this kind of involvement with the food and diet and nutrition. Anyhow, I'm just studying, or my experience is exclusively or nearly exclusively with all microorganisms that are not pathogenic microbes. Most of the microorganisms, you know, are not pathogenic. The vast minority of microbes are pathogenic. I'm actually not considering those microbes. It's another expertise. We have other experts in our department studying this kind of food pathogens. So I'm actually studying all the other microbes. And now I just want to make you a short history of my my step as a researcher. As I said, I started my PhD in 2002, and my main topic was on probiotics. And my initial part of my career was based on using, of course, classic microbiology and especially comparative genomics and biochemistry, molecular biology, all these tools for studying specific strains. And my research was pretty much reductionist, we can say, because I was trying to find some molecules, some proteins, some factors on the surface of microbes that were mediating the interaction with the host, especially in terms of adhesion and in terms of immunomodulation. And then at the same time, I was also making quite a lot of functional characterization. That means I was trying to isolate new probiotics or even studying probiotics already on the market as a work for companies. Often I did it in order to find a new possible application, new possible strains, and maybe I will make a few examples. And during this initial part of my career, I can say during the first 7, 8 years, I had the opportunity to start a collaboration which are now even the strongest collaboration that I have abroad, especially with University of Copenhagen, Toronto, and University of Tampere, where I also have a position as an adjunct professor, and so I'm often there for didactical activity and for research, especially all the part of my research regarding recombinant DNA technology is now at the moment carried out there in Tampere University. And according to my experience in the study of adhesion immunomodulation of probiotic bacteria, I used, as I said, molecular biology and this reductionist approach for identifying molecules, and this kind of activity allowed me to identify and to define for the first time several proteins and several factors, and also a novel exopolysaccharide, which are known factor now that can mediate the interaction between the host and this probiotic bacteria. And at the same time, I also made several studies of functional characterization, even basic functional characterization. And this kind of work is useful, or it was for me very useful, even if very basic in some way, especially because they gave me the opportunity to start to collaborate with the company. My research activity is quite a lot in connection with the industries, and many projects that I have are thanks to the collaboration with the industries. And this kind of collaboration allowed me to also transfer some of my strains and some of my application, and probably they are at the moment the best results that I obtain in this part of my research. So the possibility to have strains isolated, characterized, and studied in my lab, now already on the market in some probiotic products. These are just an example. And anyhow, the collaboration with company was a great opportunity for learning quite a lot, also for moving towards something different. I was probably in the best moment for studying probiotic ever, because it was around 2010, even a few years before. So it was a period of big change in Europe, because during those years, there were a couple of regulations. For example, the QPS concept was developed by European Food Safety Authority during those years. Also, the regulation on the health claims was born in those years. And that made a strong change in the industry of probiotics, and especially the biggest industries were pushed to increase research on probiotics, because in order to have a claim, now it's not anymore possible just simply using any possible probiotic, but specific studies are needed. And so I was probably in the right moment, in the right place, on the right topic, I can say. And thanks to this collaboration, then I moved my research also in what is probably now my main expertise, that is on the study of microbial ecosystems. And starting from some studies based on dietary intervention with probiotics, I first began making what are called recovery studies. So I was providing to volunteers probiotic bacteria in order to see if they can survive in the gastrointestinal tract and how they can perform in the host. Then I moved to study the impact of the intestinal microbial ecosystem. So this was the moment when, around 8-10 years ago, I started to learn how to manage bioinformatics, and so I was moving in what is called now metagenomics, microbiomics, metataxonomics, and partially now also metabolomics, so omics technology for the study of microbial ecosystems. Then I also moved to this study of impact of dietary dietary factors or dietary patterns, and I will make a few examples of this because it's now probably the main part of my research in my laboratory. And then I also have some experience now in the characterization of the intestinal microbial ecosystem in specific categories of people. And for example, my group has now a pretty nice experience in study the intestinal microbial ecosystem in subject with the irritable bowel syndrome in IBS. And finally, most recently, I also started to collaborate quite a lot with the Istituto Tumori here in Milan. And well, they were looking for a microbiologist, and I had the opportunity with them, I still have the opportunity to collaborate, to give my experience in microbes and to help them to demonstrate that the microbes that we have in our gut or in any part of our body, they have a pivotal role in cancer in different directions. For example, here I just indicated a couple of examples where For example, here on the left, we saw that the administration of microbes to the lungs through aerosol therapy have a pivotal role in determining the lymphocyte response and the immune response and reducing the formation of lung metastasis. And this kind of aerosol therapy was actually done with a simple probiotic. So the possibility to modulate immune responses with the use of safe microorganisms was very, very promising and allowed me to learn quite a lot in this field, thanks to these colleagues. And here on the right, there is an example of a recent study that has been published on animal models, on different animal models of breast cancer, where we show that the presence of certain microorganisms rather than other are of pivotal importance for determining the effect of the therapy with trastuzumab, with this monoclonal antibody in breast cancer. And so the importance of microorganisms now going all around is not only related to food and to probiotics, but they are moving also in oncology quite a lot. And anyhow, now I want to start my small seminar with an example of my studies. And I selected this one, is the so-called MAPLE project. And this MAPLE project finished a little more than 1 year ago, but we are still working on the data and trying to publish. And well, what is this MAPLE project? This MAPLE project is based principally on a dietary intervention with a polyphenol-rich diet in older people. And this project was supported by 3 funding agencies and included 3 main research groups. And the main group, the main unit was Università of Milan, and the principal investigators were me and my colleague Patrizia Rislo. She's a nutritionist from my department. And this MAPLE trial, MAPLE project, is an acronym for microbiome manipulation through polyphenols for managing the leakiness, gut leakiness in the elderly. And so I would like to tell you something about the main results that we are obtaining out from this study. Well, the MAPLE project has a general basic idea, and that is something now very well known. That means the presence of low-grade chronic systemic inflammation. Low-grade chronic systemic inflammation is, as you know, is pretty common in many non-communicable diseases, and it is also very well known for older people. And it is probably the starting point of the main non-communicable diseases which are characterizing aging. And especially the hypothesis is that during aging, there is a modification of the intestinal microbial ecosystem of the gut microbiome. Actually, not only in the gut, a very important role is played also by the oral microbiome. And the effect is that microorganisms start to be more prone to trigger inflammation and also to modify permeability. The key word in this project is actually permeability. We thought that during aging, there is an alteration of the microbiome that promotes translocation of factor from the lumen to the bloodstream, promoting then this low-grade chronic inflammation. So the idea was that it would be possible to reverse this kind of mechanism, reverse this kind of aging of the microbiome in the gut and in the mouth through a dietary pattern enriched in polyphenol, so with a polyphenol-rich diet. And so the aim of this project was actually try to have an effect on older people, otherwise healthy people, and with a dietary intervention with a polyphenol-rich diet. So the idea was to, in this kind of representation, to move this gear so that it would be possible to reduce inflammation and then improve the metabolic state and the general health state of these older people. And this project had a main part that was the intervention trial. And one of the main advantages of this project was that we had the opportunity to work and to perform this intervention trial in a very well-controlled setting that is represented by Oichi, which is a village in Padua, where older people have free living. So they are not patients, they are actually guests, and they are introduced there in several also social activities. And so they are in a perfect health condition. And there is one big advantage. And that means that people there, older people, was using mostly a couple of restaurants only, and so it was possible for us to make to control pretty well the diet. And so we made a a modification of their diet. It was pretty mild modification. We made a substitution of foods in order to increase the source of polyphenols in the diet of these older people. And this trial was a crossover trial, so a randomized controlled crossover trial. We involved older people that were otherwise healthy, we can say, independent, and the only feature that we selected as a main inclusion criterion was the presence of permeability. There are not actually valid clinical limits that we could use, so we developed our own model for defining permeability, and according to this, we selected those subjects with an increased permeability, because actually our hypothesis was that permeability has a key role in this mechanism of low-grade chronic inflammation and alteration of the microbiome. So in this crossover design study, the subject for 8 weeks consumed a controlled diet of polyphenol diets, then 8 weeks of washout, and then the opposite for each of them. And then we collected blood, urine, feces, and of course we made on this several omics analysis for out of this, especially metabolomics on feces and urine, and microbiomics for feces and also from blood. I will tell you something about this kind of pretty tricky experiments on blood. And then, of course, evaluation of food intake and anthropometrical, physical evaluation. The diet was not strongly modified. We just made a substitution of some foods, and we selected from industry high-quality products with a characterized and well-known high content of polyphenol. And here there is just an example of the table of the single meal of what we introduced in the meal. So there were 3 doses of polyphenol sources, as you can see. And the main source of polyphenols that we used were cocoa powder, for instance. It was actually the food with the highest content of polyphenols. Then Renetta apple on blueberries, and pomegranate juice, and also blood oranges and juice, and also green tea. Yeah, these were the main foods that we used that allowed us to obtain on average for all these other people an intake per day of 750 milligrams of polyphenols. So it is, even according to nutritionists, recommended amount of polyphenols is not possible to increase too much, because they could even have, especially in the elderly, some detrimental effects. So we decided to use this kind of amount. If someone of you has experience with polyphenols intake, this could sound something like not so high, but actually, according to the baseline, it was a significant increase for twice or even 3 times more than before. Then we performed this analysis, this trial, and we assessed at the beginning approximately 500 older people, and out of those we selected, according for increased intestinal permeability, 51 subjects that were able to conclude per protocol the study. Such a kind of numbers could sound pretty small, but it's not like that, especially in human intervention trial in so-called applied nutrition in crossover design, 51 subjects. It's absolutely more than enough, according to literature, for testing our hypothesis. And the first publication that we did, we're just summarizing the main result, the first results that we obtained. It was published in 2021, so quite recently. And just summarizing, we immediately observed 2 main results. And the first result was that, fortunately, the main endpoint, the primary endpoint of the trial, was reached, because we found a significant reduction of zonulin, that is a marker of intestinal permeability, is the main marker that we used, together with another that I will describe in a while. And together with that, we also observed a very clear modification of the intestinal microbiota in the polyphenol-rich diet compared to the control diet, especially in terms of increase of fiber-degrading and butyrate-producing microbes. Among these microbes, one of the most important are Butyriccoccus and Fecalibacterium prausnitzii, that is a well-known anti-inflammatory bacterium. It is known to produce butyrate, that is an anti-inflammatory molecule, but it's also important for the production of certain peptides, which has been demonstrated to be anti-inflammatory in the gut of everyone. And this microbes is now very much studied also for a potential application as a next generation probiotic or live biotherapeutic more properly. And then we move forward for more in-depth analysis, and especially with the omics. And I want to summarize here very quickly what we found thanks to metabolomics. We perform metabolomics, targeted and untargeted metabolomics on serum and urine. And the analysis of serum was very interesting because it was very clear the result and concerning how certain metabolites that derive from polyphenols are a key element which are able to connect permeability and certain intestinal bacteria. Especially we found two molecules which are theobromine and methylxanthines. Theobromine are very commonly found in cocoa. The main source of theobromine is cocoa and chocolate. And methylxanthines are on the opposite, mainly in green tea. And especially these two molecules were found to be very well connected and correlated actually in a direct way, in a positive way, with the same bacteria that were enhanced by the dietary intervention. So with the butyrate producing bacteria and fiber degrading, such as Butyriccoccus and Fecalibacterium, as I said before. And at the same time, these same molecules were also inversely correlated with zonulin. And also a network analysis evidenced the existence of this kind of link between the metabolites of polyphenols, certain gut microbes, and zonulin and permeability. And these same results, or actually we can say the importance of permeability, was also very clear when we made metabolomic analysis on urine samples, because the analysis of urine suggests us that only subjects with a healthier intestinal integrity, so with a lower permeability, were able to properly metabolize polyphenols. Exclusively those subjects with intact or less altered, we can say, with a lower permeability, were able to modulate, to transform the polyphenols in those molecules which are the most health promoting molecules. And of course, this kind of mechanism seems to be connected to an alteration of the microbiota that could be partially reverted by the intervention with polyphenols. Anyhow, once that we found these initial results, we understood that actually permeability could be a key element for understanding this effect that polyphenols can have on the host in relation to the microbes in the gut. And so we decided to start the analysis also of blood samples to go more in depth on permeability. And we decided in this project to spend quite a lot of time in metagenomic and microbiomics and metataxonomics, so molecular biology applied to blood samples. So in a few words, we try to define and to detect and define which are the bacterial DNA present in blood. This kind of activity and this kind of work is pretty tricky, and at the beginning we were pretty scared about the possibility to obtain wrong results. I mean, during those years, there was the experience of people working on and publishing data concerning the presence of bacterial DNA in placenta, human placenta. That kind of results were very exciting, very interesting. But after a couple of days, it was also demonstrated that those results were fake, actually were wrong, because it was just because of contamination. So we were afraid to make the same mistakes. So to find something in blood that is actually just from contamination. So we spent quite a lot of energy and approximately a couple of years to find a proper protocol in order to be sure or to convince ourselves that actually it's possible to detect the bacterial DNA in human blood. Of course, bacterial DNA in human blood is very low amount, much less than what you can see as compared to the human DNA. So that is the tricky part. And what we saw is actually that it's impossible to avoid contamination with DNA once that you perform this microbiomic analysis. But at the same time, we obtain even some encouraging and some stimulating results, because anyhow, we always found in all these blood samples, and we tested hundreds of blood samples, a level of bacterial DNA that is always higher than our controls. And probably what convinced us that probably something true is this kind of results that we saw on several population of blood samples. That means a correlation between the level of bacterial DNA, bacterial DNAmia, we can call it, in terms of 16S ribosomal RNA gene copies, because we made this quantification simply with quantitative PCR. So a correlation of this bacterial anemia with zonulin, so with the main marker that we use for intestinal permeability. And this correlation means that, I mean, it makes sense. Every time that there is an increase in permeability in the people, there is also the possibility to find a higher presence of bacterial DNA in blood. And this is very clear in the older people. We obtained this in a different population of older people. And we also made a taxonomic profiling of the DNA that we detected in blood. And we saw that virtually all the DNA, the bacterial DNA, is circulating in the blood of these older people, but in general, I can say also in adults and healthy people, or healthy adults, belongs to only two divisions, proteobacteria and actinobacteria. And it was another interesting result, because actually it's something that we can find also in literature. Independent studies made with similar approaches found the same. And also, if we give a look to clinical data, especially those experiments made cultivating blood, cultivating the microbes which are alive in blood of people in the absence of sepsis, and they actually are in some way in agreement with our results. And especially, we found one genus, which is Pseudomonas, this gram-negative bacterium that was the most commonly found, I mean, the most prevalent, and especially certain taxonomic units within this genus, especially taxa related to the group of Pseudomonas fluorescens, and not aeruginosa. Pseudomonas aeruginosa is a typical nosocomial pathogen that gives also often a blood infection. But actually, what we found was not this kind of pathogenic bacterium, but other Pseudomonadaceae traces of DNA that are more closer to fluorescence. And this is, again, in accordance with clinical data. And for this reason, we decided to use this protocol that we developed in this study, also in the MAPLE study, in the MAPLE trial. And so we analyzed all the blood of these people involved, in these older people involved in the MAPLE trial. And we decided to make this kind of analysis. That means stratifying people according to the level of bacterial anemia. We then found something interesting, because we saw that very clearly older people with an increased level of bacterial DNA in blood were also characterized with an altered metabolic state, also in terms of increased amount of triglycerides and lower amount of HDL cholesterol, and increased amount in blood of tumor necrosis factor alpha. So actually, this was something like a marker in these older people of a worse metabolic and immune state for this patient. And what we then also observed is that in the same patients, when we analyzed the effect of the dietary intervention with polyphenol, the best results that we obtained were observed exclusively in those people with an increased bacterial anemia, especially in terms of reduction of zonulin, and even a clear reduction of interleukin 6 was found exclusively in subjects with an increased bacterial anemia. And so this was in some way a suggestion that probably this analysis, this data and this measurement of the bacterial DNA in blood could be an interesting marker for selecting the part of older people that could most benefit from a dietary intervention like the dietary intervention with polyphenols. So as a partial conclusion, then with this study, we demonstrated and we observed that the initial hypothesis was right. That means that intestinal permeability is probably one of the main elements. We do not have yet any valid clinical method for making a measurement of intestinal permeability. In general, in hospital, they use this assay that is based on the analysis of sugar. It's called sugar test. So after the consumption of sugar, there is a measurement in urine, but it is pretty difficult to perform. It takes longer, and especially in older people, this is not properly a proper method. And so permeability should be a hot topic for the study of this metabolic condition in older people. I can say in general for all the research on non-communicable diseases. So this project actually evidenced this, highlighted the importance of permeability in connection with the intestinal microbiota. And then we also found the importance of the intestinal bacteria and how they can so strictly important in defining the effect that an intervention with polyphenols, or in general polyphenols, the effect that polyphenols can have on human health. Different microbes in the gut means that we can obtain very interesting positive effect from the polyphenols, or the opposite. So there is a huge inter-individual variability depending on the microbes in the gut for the polyphenols, at least. And finally, we also proposed this new potential marker. We found this circulating bacterial DNA as an interesting potential marker, and the ones that the protocol is demonstrated to be actually free of contaminants, and could be, it is now applied. We are trying to apply this also in other projects, especially one that is ongoing on blood samples collected from around 500 colorectal cancer patients. We are trying to see if there is some signature in the blood of these patients, and so we are using the same protocol. More in general, if you want to take even some lecture from this experience of our MAPLE trial, and even according to literature, one take-home message is that, as I said also at the beginning, we can better understand which is the relationship between food and human health if we consider microorganisms, and especially microorganisms associated to the human body. And I also said, even if I spoke mostly about gut bacteria, also oral bacteria, skin bacteria, vaginal bacteria, they have a strong importance as well. And literature is very interesting now in this field. We have many examples where we can see that we can properly understand the mechanisms linking certain nutrients with human health, exclusively if we consider the intestinal microbiota or microbes in general. Here I just indicated a few examples, which are probably the best known. There are interesting studies demonstrated that milk fat can have an effect on colitis in a way that is mediated by certain gut bacteria, such as Biophila Wadsworthia. Another very interesting study is this one that is connecting the effect of high salt diet with hypertension, with a mechanism that is linked to the presence of lactobacilli, of certain lactobacilli in the gut. The absence of those lactobacilli can increase quite a lot the effect of high salt diet and salt on hypertension. Other interesting and very well-known examples are represented by dietary fibers and carcinogenesis in colorectal cancer, and so on, and several others. Anyhow, the last example that I want to give you is related to this one in the middle, and it is probably the best example because it's now very well known. And out of these studies, now we have a potential new clinical marker that has a clinical value that is represented by TMAO, that means trimethylamine oxide. And these kind of studies were very revolutionary in some way because they were showing that atherosclerosis and the level of cholesterol is absolutely connected to diet, but it could be independent, completely independent by the intake of cholesterol. In these studies, it was shown that the intake of molecules like carnitine and choline, especially in the form of acetylcarnitine and phosphatidylcholine, they are essential nutrients. We have quite a lot. We introduce grams of these molecules every day, every week, I can say. And they are a rich source of carnitine, and choline are, of course, animal products, but also in vegetables, choline is quite a lot present. So we introduce a lot of these molecules, and we need it quite a lot. It's an essential nutrient. They are essential nutrients. But once they reach our gut, it depends quite a lot by the microbes that we have in our gut, because several microbes can convert this molecule in trimethylamine very easily, this kind of conversion, and several bacteria can do it. And this kind of conversion anyhow produce a molecule that is toxic. Trimethylamine is rapidly absorbed, and once in the liver is converted to trimethylamine oxide, and trimethylamine oxide is then a toxic molecule that is well demonstrated to increase through several mechanisms the level of cholesterol in the blood. And what is pretty interesting, and also our contribution with this paper is telling this, is that there are some microbes that have this kind of gene, the TMA liase gene, for example, CUT-C is the name, especially in gram-negative, in Enterobacteriaceae, which are very important in the production of trimethylamine, of this toxic molecule. But what is clear is that the level of these microbes is very much variable from subject to subject, especially according to dietary pattern. For example, we observed also together with other research groups that the dietary Mediterranean diet can reduce the amount of this gene, of this bacterial gene in the gut, and so also the level of trimethylamine oxide. And at the same time, other diets, especially so-called Western diet, particularly rich in animal products and lower in fiber, we always know this, they can increase quite a lot the level of this gene. So this kind of experience, that all these examples that I have indicated here are telling us that studying microbes could be also very useful for understanding this inter-individual variability, and we can allow us to have some tools for creating what is called personalized nutrition. So trying to find the proper diet, the proper nutrients for specific subjects. And this probably the main source of variability in terms of the effect of nutrition is not connected to the genotype of the people, but is connected to the microbes that we have in the gut. And the microbes that we have in the gut, of course, they depend by the diet, by our dietary habits, but it depends quite a lot by the first state of age, for the first years of our age. So what happens when we were born until 3 years old. And so understanding which is the link between certain microbes and the effect of diet, of course, could be the future for a more personalized nutrition. And so with this final message, I want to conclude my presentation, just telling something about the people that is working with me at the moment in my group. We are in the moment, beside me, other 7 people, and especially I have 2 researchers. Valentina, she's at DB, and she's working especially in all the model of interaction and modern cell line models and interaction with the host. And then Giorgio Gargari, which is my bioinformatician and data scientist, so it's a key element now because all the omics need not so much wet science, but especially computational science. And then the last one that I want to mention is Giacomo, that is actually the microbiologist, my alter ego at the moment in the lab, and several other people here. And so thank you very much. And if you have any question and there is something that attracted your attention, your interest, of course, just be free to ask me. And thank you.
Hi. Ciao. Beautiful work, really. I really like the idea behind the MAPLE study, because really doing the dietary intervention studies is not easy at all. I was just wondering if you were also able to somehow control for the already present comorbidities of these patients that might have generated the permeability of the gut.
Sure. And we also consider drugs because during all the trial, there were also a group of physicians that were the physicians working there in that village in OEC, and all these people, so they were actually well controlled, and we had a list also of drugs. We removed an exclusion criterion, for example, was the consumption of inhibitory pump inhibitors or other gut problems that requested a high consumption of drugs that can reduce the intestinal acidity, because it is connected to, of course, to altered permeability. We also removed, we don't include people with diagnosed state of irritable bowel syndrome, and because that one is a condition that is associated with, of course, intestinal altered permeability, and also with SIBO, because some of them, they also were removed because they had some analysis of gas tests, so they were increased altered emission, so they were diagnosed with SIBO. So also at least 3 older people were removed because they had also SIBO. So we try to take into consideration this condition. Of course, they are older people. Some of those were over 80, over 85, a couple of them also over 90 in excellent, I have to say, health condition. And they were consuming drugs, and so other medication were not excluded at all. So, of course, the group is quite heterogenic. Also, in terms of permeability with the markers that we used, there was quite a lot of variability, and it's virtually impossible to avoid this kind of variability. Yeah, we considered gender, and we found some difference in terms, I hope to remember. We had a better effect of diet intervention, especially for certain metabolites of polyphenols in women rather than men in terms of inflammation, and in men rather than women in terms of triglycerides and lipidemic markers. So we found this kind of difference, even if the group was pretty small, and I mean the group of subjects. So we were actually quite surprised. And according to permeability, we obtained significance both with male and female, even if significance was higher with females. And in general, to my experience, I have to say, when we make some dietary intervention, especially when these interventions are not with a specific nutrients, but there is something like a dietary pattern that we want to propose, the best, of course, also the best compliance, but I have to say also the best results are with women. I do not know why. Probably it's because of a better compliance, of course. They are more serious. I don't know, but if we remove compliance, I don't know why, but often with women, we have better results. It seems that they can answer the response, the metabolism can respond better to these dietary patterns, especially in older people. And so just very general, probably is not very scientific answer to this last part, but actually it's my impression at least.
Thanks a lot, Simone, for this presentation. And this is just, I mean, it's not really a question related to what you showed, but it's just, I mean, your idea and philosophy. And you show that a lot of depends on the variability that you have and the interaction with the host and with the microbiota and so on. And at which point are we in understanding also the contribution of the pangenome of the microorganisms that you have? Because now we are understanding more and more, and how this difference is also taken into account and how can influence.
Yeah, well, the amount of data now are really enormous. There are so many, and we need now more time or more tools for managing the data rather than creating other data. And the pangenome for the intestinal microbiome is so huge, it's enormous. You can find genes virtually for everything. So if some people is making this kind of gene fishing for certain enzymatic activity, probably one of the main examples that I can make is related to people in Japan that were found with some ocean genes for the metabolization of porphyrin agarose. They are present only in Japanese people. They were acquired in some way by their microbiome, and is not present in maybe in Milan people, yes, because we are using now to consume so much sushi, so we also acquired those microbes. And so there is huge variability, and the point is that there is also huge variability in the ratio between microbes. I mean, also butyrate producing bacteria are present in every one of us, also in sick people. There is not an absence. It's just a matter of ratio among these bacteria. And so it's a very complex system. And at the moment, trying to find what I said before, so a personalized approach is virtually impossible. It's very difficult, but it is also true that maybe for certain applications which are not for healthy people, but in specific disease or and illness states, actually there are some initial important and possible application. Also, my experience with the colleagues working in the Istituto Tumori, they are making really excellent experiments, and what they are able to find in terms of the response to treatment with trastuzumab, this monoclonal antibody, it's very impressive. In mice, it's really incredible. I mean, you change the microbes in the gut, you increase Clostridiales bacteria, or they are called now Eubacteriales, and you decrease other microbes, or you increase gram-negative, and you have absolutely the opposite. The response of trastuzumab could be completely absent or very present. And now they are trying to find something similar in patients as well. And so they are now translating this also in humans, and probably the main first interesting results are there in the trying to define how the microbiome can influence the effect of the treatment of specific drug treatment so with patients rather than healthy people. In nutrition, its variability is so high and so difficult to find the right topic. Yeah, thank you.
I have just a curiosity on people that follow this diet, like vegan or whatever diet. If the microbiome is so different, if their response is to...
Yeah, there are many studies already showing a modulation of the microbiota according to certain dietary pattern, vegan rather than vegetarian and Mediterranean and so on. Anyhow, it can be easily found. I mean, you can even find a modulation of the microbiota, that changing the microbiota, even with a simple dietary intervention with probiotics. So something very simple. And so you can imagine if you change your dietary pattern, the change is very evident. And the point is that we know and we have the definition for eubiosis and dysbiosis, but we do not know exactly what is a precise eubiotic microbiome in the gut and what is a real dysbiotic microbiome. And so we have definition. We know that it's something that is can be shaped pretty well, but we are not yet at the point to say that the modification of the microbiota with certain dietary patterns is for sure detrimental rather than not, with a few exceptions. What is very clear, for example, there are already several studies. Some of those, probably the best studies in this field, are made by colleagues in Napoli, the group of Danilo Ercolini that was studying Mediterranean diet, and they show very well that several metabolic activities, microbial metabolic activities, rather than specific taxa, for example, also this level of the TMAO or the production of butyrate, they are much better with the Mediterranean diet, rather than vegan, rather than vegetarian, rather than, of course, Western diet. So we already have some indication in that direction. And at the same time, another very clear idea is connected to the so-called large hygiene hypothesis. That means that the chronic intake at a too low level of fibers is promoting low-grade chronic inflammation due to the enhancement of certain microbes in the gut and the reduction of butyrate-producing microbes in the gut. This is another thing that is very well known, and it is for sure connected to diet, at least through longer period of time. Yeah, but actually, I believe we are still at the beginning, even because most of the tools, even what I have done, is at the level of taxa, and it is a strong limitation. With this kind of studies, we didn't have money enough, and at the time also probably competencies for making metagenomics, that means shotgun, and analyzing all the DNA, or maybe also a very precise metabolomics is pretty difficult. And taxonomy and taxa are so variable from subject to another that sometimes you cannot get anything out because of this high variability that is not connected to variability in function. There is this concept in all the microbial ecosystem that is called functional redundancy. That means in terms of taxonomy, two ecosystems could be very different, but in terms of functions, they could be very similar. And the typical example is with butyrate. Many people here is probably characterized by many Faecalibacterium prausnitzii as a producer of butyrate. In my gut, I know my gut Faecalibacterium is very low, but I have high butyrate thanks to Roseburia. So different taxa, same function. So this functional redundancy is limiting quite a lot the efficacy of study this topic with metataxonomics. We need to move to metagenomics, but you have to put 1 0 more in the cost of your experiment. So we are moving in that direction, but it takes time. it takes money and time, and it's getting cheaper and cheaper. So probably in the near future, we can move more quickly in that direction.
Type of studies that they call like transferring the blues. So that's something that people take the microbiota from people suffering.
Ah, you mean the microbiota transplantation, fecal?
Yeah, fecal transplantation, but they can transplant some pathologies like.
Ah, sure. Yeah, is something that is
depression.
Sure, yeah. Because non-communicable diseases are strictly connected to the skewing of the immune system that is mediated by the gut bacteria and the overall community of microbes there. And when you transfer the microbiota from one subject to another, you transfer also the predisposition to this kind of diseases, and you cannot know before. And so there are a couple of examples, even more, very clear. One is connected to obesity. It was young women with problem with the Clostridiales difficile recurrent infection. She received from the sister, overweight sister, the microbiome, and then he was lean and became obese. And another example was even transfer of this problem, health problem and depression and so on, yes. Well,
but are there any mechanisms known?
Yeah, some mechanisms are already known. I mean, the link between central nervous system and the gut is very well known. There are many studies. Well, there are even some applications. For instance, in South America, it was pretty common to treat certain depression condition, even severe depression, with some worms, actually, with some pathogen and worms given to people. So creating an infection in the gut, so creating a certain kind of disease in the gut in order to solve the problem of depression and the central nervous system. So the existence of this kind of axis is very well known, and the mechanisms are at different levels. One of the best known is that there is a huge production of neurotransmitters that are in the gut. If you think about all the metabolites derived from tryptophan, the availability of serotonin, 90%, 85% of serotonin is produced in the gut, and this kind of production is connected also to the activity of microbes in the gut. The carboxylase activity on tryptophan is very common in gut bacteria. Some of the gut bacteria, they can even make the transformation of tryptamine into hydroxytryptamine, so serotonin. So there is also this kind of involvement. And then there is also an involvement of the vagus nerve. It was shown very well in mice models, especially with bifidobacteria, that the effect that they were able to see in the central nervous system once that they administered the bifidobacteria can be removed after resection of the vagus nerve. So also vagus nerve is involved in this. And of course, the immune system is involved because actually the low-grade systemic inflammation that is common in all these diseases is strictly connected to the gut bacteria and to several features of the gut microbes and the gut ecosystem. For example, the reduction in biodiversity that is typical of all the people living in Western countries. We have a microbiome that is much more poor than people living in a rural area or in countryside, and probably also people living in Milan has a lower diversity. And this kind of lower diversity in some way, it seems like the immune system cannot be managed in the proper way. And so as a consequence, there is this alteration of immune system, enhanced presence of inflammatory factors, and those factors, of course, they have an effect of the central nervous system as well. It's incredible how all these diseases are so much well connected together. The condition called metabolic syndrome is at the basis of virtually all these diseases: obesity, NAFLD, so non-alcoholic steatosis, and diabetes, certain kind of cancer. They have at the basis the same metabolic alteration that can be called metabolic syndrome. And this metabolic syndrome is strictly connected to this low-grade chronic inflammation and to this alteration of the gut microbiota. So this something that is probably in the same area.
So if you have inflammation in depression, then you can cure depression with anti-inflammatory drugs.
Why not? I'm not sure if it is done or not. I'm not an expert about depression, but a link is present. I mean, if you think about the comorbidities between gut function and depression also, and several drugs that are given for central nervous system problems, Prozac, for instance, is very much affecting the intestinal motility because the target is SERT, that means the receptor for reuptake of serotonin, and so these kind of drugs they are modifying quite a lot the functioning of the gut. At the same depression from mild to even severe has a high incidence in people with inflammatory, of course, inflammatory bowel disease, but also in irritable bowel syndrome. IBS patients, they often have this kind of problem connected to the central nervous system. So there is this kind of comorbidity, and I believe that inflammation is involved. It's probably not high-level inflammation. It's not acute inflammation. It's this, as I said, low-grade chronic inflammation.
Okay, other questions? Okay, if not, thank you very much.
Thank you very much to you, and thanks.